Okay, so hello everyone. My name is Maria Teresa, and I will present you my thesis, The Effect of Food Quality and Ocean Acidification on Fatty Acid Composition and Gene Expression of the harpacticoid copepod, Platychelipus littoralis. Okay, so due to anthropogenic activities, the rise of greenhouse gas emissions, such as CO2, are increasing not only the global mean temperature, but are also causing the acidification of the oceans, as they can absorb the CO2 from the atmosphere, inducing chemical changes such as the decrease of the pH. Actually, the global ocean pH is around 8.2 units, although it fluctuates due to seasonal and regional variations. However, the pH has already dropped 0.1 units since 1850, and it's expected to decrease around 0.5 units more by the end of the century. Ocean acidification affects growth, reproduction, and behavior of marine organisms, and there are some organisms that are more affected than others. For example, copepods, they are being seen like resilient to ocean acidifications, and these are small crustaceans that are the major component of meiofauna after the nematodes in marine ecosystems. They play a crucial role as they transfer energy from primary producers to higher trophic levels. They have high nutritional values, as they have high concentrations of fatty acids. Therefore, understanding how ocean acidification affects copepods is crucial to assess the impact of how nutritional values could affect higher trophic levels. So these fatty acids are components present in all living organisms and are essential in many physiological processes, like the omega-3 essential fatty acids like EPA, DHA, and ALA, which are essential polyunsaturated fatty acids called PUFA, and they are involved in reproduction and somatic growth of fish larvae. However, these PUFAs need to be obtained through the diet, as most of the animals cannot really efficiently synthesize these fatty acids. And here, microalgae plays a crucial role, as they're abundant in lipids. So this Platychelipus littoralis is a benthic harpacticoid copepod, which is dominant in the top sediment layer in intertidal mudflats, and it can elongate short-chain fatty acids and produce PUFA. So, and this ability is called bioconversion, and this bioconversion makes this copepod very special, as this ability is not shared among all copepods. So it is suggested that they contain fatty acid desaturases and elongases that are involved in this process. So the overall aim of this thesis was to investigate to what extent the changes in the fatty acid composition and expression of certain genes involved in PUFA bioconversion take place in a population of Platychelipus littoralis exposed to ocean acidification and fed with high and low quality food. The hypotheses were that the copepods had the ability of fatty acid bioconversion, that a low-quality diet and ocean acidification affect the fatty acid composition of the copepods, and that a low-quality diet and ocean acidification affects the expression of these PUFA bioconversion genes. So around 2,000 copepods were sampled from the Paulina intertidal mudflat located in the Western Kelde estuary in the Netherlands. After the field collection, 200 individuals were cleaned, and they were stored at -80 in order to be used later for fatty acid analysis, to use them as a control. And the rest of the copepods were kept overnight without food, just to acclimatize them and empty their guts prior to the experiment. So for the food sources, the algae were obtained from the algal collections of the Ghent University. For high-quality food, we chose the diatom Nitzschia, as it is rich in PUFAs, and for low-quality foods, the chlorophyll Dunaniella tertiolecta, as it is poor in PUFAs and it lacks of EPA and DHA. To prepare the incubation water of the treatments, so I had a stock of acidified seawater, which I previously bubbled with CO2 gas, and I used this stock of water to prepare daily the controlled seawater, which was around pH 8, and it was the pH where the copepods come from, and also to prepare daily the acidic seawater, which was 0.5 units of pH less. So 110 copepods were in each experimental units. There were the two different okay, two different pH. Oh, it doesn't work right. Okay, two different pH. The two different diets, Nitzschia and Daniela. They were for replicates per treatment. They were in a constant temperature, which it was similar to when the copepods were sampled. And they had a light and dark cycle, and they were exposed under the treatment for 10 days. And after the 10 days, 50 copepods per each experimental unit were randomly picked and taken out in less than 7 minutes, and were flash frozen in liquid nitrogen, and then they were stored at -80 in order to be used for qPCR analysis. And another 50 copepods for each experimental unit were left overnight. They were clean and they were left overnight without food, but under the same water treatment in order to empty their guts, and then next day they were stored at -80 to analyze their fatty acids. And these fatty acids were analyzed using a gas chromatograph with a mass spectrometer. In order to perform a qPCR, we designed primers from the transcriptome of the copepod Platychelipus littoralis. And the target genes were the genes that we were focusing on. These are genes encoding putative, desaturases, and elongases. And the housekeeping genes are genes that need to be included in qPCR analysis, and their gene expression are not really affected under experimental conditions. Total RNA was isolated from the copepods, and complementary DNA was synthesized from it. An RT-qPCR was performed using the fluorescent dye cyber green to measure the gene expression. There were triplicates per sample and primer, and also negative controls were included where they didn't have the cDNA as a template. So these are the software that I used. I perform a one-way ANOVA for copepod survival, and for the total fatty acid content of the copepods that were not exposed under the treatments, and the copepods that were exposed for the treatments. And the two-way ANOVA to evaluate the effect of diets, pH, and their interaction in the fatty acid composition and gene expression of the copepods, and multivariate analysis in the data set of fatty acids. So there was an overall high survival of the copepods regardless of the different treatments of pH. There was a significant decrease of the total absolute fatty acid content at the end of the experiment compared to the field copepods. And this could be explained because in the field, the copepods can fit on a variety of diets. And here in the experiment, we only fed them with one diet, so a decrease could be expected. So there was no negative effects of diets. However, if you remember, Duranella tertiolecta lacks of EPA and DHA and is very poor in omega-3 fatty acid. However, you can see that the copepods that were fed with Duranella tertiolecta under control the treatment seem to efficiently bioconvert these fatty acids. And also the copepods that were fed with Nitzschia seem to efficiently bioconvert this fatty acid, which was also absent in the diet. However, studies suggest that fatty acid analysis should be combined with compounds-specific carbon isotope analysis labeling the food sources in order to support these findings, to support this fatty acid analysis. There was no strong effect of ocean acidification in the fatty acid content. However, there were 24 fatty acids detected in the copepod, and only one fatty acid, which is oleic acid, was affected by acidic pH. So first, one of the reasons why we didn't really see a strong effect of ocean acidification could be because we gave the copepods food ad libitum. This means that a lot of food was given to the copepods. And studies have shown that copepods can increase the food intake in order to compensate the extra energy demand under acidic conditions. Another reason, because maybe the experiment was too short, it was 10 days, so maybe it was not enough time to see an effect, a negative effect. Also, these copepods live in intertidal mudflats. They live in very dynamic habitats, so maybe they have adapted to fluctuations of pH. And also, studies have shown resilience of adult copepods to ocean acidification, and in this study, we only used adults. So in this MDS, we can see a clear separation of the food sources, which had, of course, very different fatty acid composition. And we can see a clear grouping of the copepods, both field and the copepods under the treatment, because at the end they have quite similar fatty acid composition. And it can also be seen here in the simple results where, regardless of the pH treatments, the similarity of the fatty acids was very high, was very similar between them. So regarding the PUFA bioconversion genes, we found a high expression of Δ5, Δ5, Δ9, and elongase 2 under the tertiolecta, tertiolecta diet under current pH compared to the ones that were fed with Nitzschia. You can see this like high upregulation. So this means that low quality food induced this PUFA bioconversion, but by synthesizing these missing fatty acids in order to compensate for this lack of fatty acids. Also, we saw a high expression of the of the gene Δ5 under both diets under the acidic pH compared to the controls, as you can see. However, of this like very clear upregulation, none of the fatty acids that result from a Δ5 desaturation were affected. And also, regarding the Δ9 gene under the so there was a higher expression of the copepods of the gene Δ9, sorry, under the Nitzschia diet under the acidic pH if you compare them with the control treatment. And actually here a Δ9 desaturation produces it's in charge to produce oleic acid. And oleic acid was the only fatty acid that was negatively affected by ocean acidification. So you can see that maybe the acidic pH affected the synthesis of this fatty acid. So you can see that. So to conclude and to bring you back to the hypothesis also, we saw that the copepods show the ability to bioconvert short-chain fatty acid and produce PUFA when they were fed with low-quality food. Also, the low-quality diet did not have any effect on the fatty acid composition. There was no strong effect of ocean acidification in this fatty acid composition. And as I said before, only one fatty acid of 24 was negatively affected. Regarding the PUFA bioconversion genes, we could see that low-quality diet induced PUFA bioconversion by significantly increasing the expression of the fatty acid desaturases Δ5, Δ9, and elongase 2. And also we saw a high expression of Δ5 and Δ9 genes under ocean acidification. A recommendation for future studies will be to include also another naturally occurring stressor, such as the increase of the pH, the increase of temperature, in order to have more accurate predictions of how copepods will be affected by climate change. Also, it will be good to expose the food sources under the same treatments as the copepods. And finally, this analysis, fatty acid analysis and gene expression analysis, should be also studied in earlier developmental stages like NOPLI. Finally, I would like to thank my supervisor, Jens Boyen, my promoter, Marlene de Trocht, and everybody that was part of this thesis, and thank you all for also coming and listening.